Artium with a difficult last name.
You don't
Artium.
need to pronounce the last name. It's too
Yes,
long.
please, how do you pronounce it?
Khatchtouriants.
Exactly what I wanted to say. And he's going to tell us about the different projects with a very, very interesting instrument that we have in the Nano Center, this combined atomic force microscope, scanning probe microscope, confocal microscope. And Raman.
I would like to thank organizers of this event for giving me the opportunity to give this presentation. Probably it's better. Okay, works. So again, sorry for the unexpected delay, and I would like to thank the organizers of this meeting for giving me opportunity to make this presentation. Unlike other people who presented their presentations on this meeting, I'm not conducting my own research, but I'm rather helping other research groups in their research with the equipment which is available in the laboratory I'm responsible for. And this is biological AFM and Raman spectrometer. So I will present a short review of current research projects I am involved in. These are not all the projects, just selected ones. So let's begin. As you know, in our Nano Center there is a relatively new laboratory. It's already not so new. It's a little bit more than 1 year. Which comprises two new state-of-the-art systems. One is the AFM. You see here the head of the AFM of the JPK company located in Berlin, Germany. This is the AFM head. And here you see the Raman spectrometer. This is the spectrometer, and the AFM sits on the inverted microscope and is fully integrated with the Raman spectrometer. And there is also another microscope, which is the upright one for only Raman. And these systems are quite modern and are equipped with many features. Like all modern systems, they possess all the features like resolution and the noise level in AFM. In particular, this AFM has an ability to scan in liquids, and it is designed to work with biological samples. So it has a large tip scanner, 100 over 100 over 15 microns. It has many scripts and algorithms in the software, allowing to work with biological samples to acquire their mechanical properties. For example, it also has the direct overlay feature, which allows you to import the optical image, bright field or fluorescent image, into the AFM and to correlate it with the tip position and to directly move the tip to the desired area and take AFM scan there or probe the mechanical properties. And it is also equipped with a stage with the temperature control from 15 to 60 degrees centigrade, which actually covers all the range of required for biological samples. The Raman system has 3 lasers, 532, 633, and 785. It is capable of taking Stokes and anti-Stokes Raman scattering, anti-Stokes for the 633 line. It has quite good resolution because of its focal length of its spectrometer. The resolution can get up to 0.3 centimeter in -1. Achromatic design, mapping option, also fast mapping option, and it also has a heating cooling stage working from the liquid nitrogen temperature up to 600 degrees centigrade. Now we are going to projects which I mentioned. One of the projects is in collaboration with the research group of Fernando Patolsky. They build self-assembled monolayers of lipid DNAs on the silicon oxide and the gold surfaces. Such self-assembling monolayers are of great interest because of their remarkable properties in terms of like electrical properties, chemical properties, optical properties, and then they further functionalize the surface by attaching so-called amphiphilic DNAs, which have hydrophobic tails, and they are just anchored in the self-assembled monolayer of the lipid DNA. And this process appears to be controllable by the incubation time, the density of the concentration of the amphiphilic DNAs, and also reversible, which is very important. By just heating it to 80 degrees centigrade, one can release these DNAs and to stay with the original self-assembled monolayer without compromising its original properties. So one of the important tasks in such research is to characterize such surfaces, and they are characterized by different techniques, including fluorescent confocal microscopy, if you link some fluorescent dye to them, or they are characterized by X-ray techniques. And also, it appears that it can be characterized with AFM. At a first glance, there is not too much to do here with the AFM because these monolayers, they're quite thin, and attaching these molecules is not expected to change their surface roughness, for example, too much. So the height channel of the AFM is not going to give too much information. But in taping mode AFM, as you know, there is also phase option, and the phase is very sensitive to the interaction forces between tip and the sample. Here you see the pattern of the sample-assembled monolayers, where you see the green lines and the green squares. They are approximately, they're quite big, it's 30-35 microns. These are lipid DNAs here, and on them we anchor the amphiphilic DNAs. So you see here the phase image of the AFM before and after anchoring the DNAs. So you see here that here there is just a hardly seen contrast, but here the contrast is quite prominent. So in this case, it gives additional information about the sample. The next project is a project with Dr. Dan PR from Life Sciences, and they are trying to functionalize cell lines of macrophage mice cells with single-wall carbon nanotubes. So you know, single-wall carbon nanotubes are of great interest also in biology. Because they can help in drug delivery and then for diagnostic purposes, but it is difficult to use them directly in biology because they first are insoluble in water because they are highly hydrophobic, and also because they are quite toxic in their intrinsic form. So you need to functionalize them. So in this research, the raw carbon nanotubes were linked to a molecule of hyaluronan using phospholipids, which have hydrophobic tails. So you have this assembly, which is then directed by specific techniques to the membranes of the cell lines to be investigated. And here we used Raman for the characterization of these samples. As you know, carbon nanotubes have a very well-defined peak at about slightly less than 1,600 centimeter minus 1. And then, for example, on this spectrum, you see the spectrum obtained from the membranes of these cells and from the interior of these cells. They're quite similar, except that on the membranes we see characteristic peak exactly on the position where single-walled carbon nanotubes peak would appear. We also took control cells where there was no functionalization with carbon nanotubes, and we can see that on the control group this peak doesn't appear. So we can conclude from this that indeed the functionalization of cells with the carbon nanotubes succeeded and that they are primarily attached to the membranes. The next project is the project with the group of Professor Eliezer Giladi, and it relates to electrolysis and electrolytic properties of rhenium nickel containing alloys, and these alloys, they have quite remarkable properties, as also mechanical and thermal stability as well, and they are of interest for research. And it is known that if, for example, in electrolytic solution only nickel is present without rhenium, or vice versa, then the coating efficiency and the electrolytic efficiency is quite poor. But if one adds nickel, some nickel concentration on the rhenium solution or vice versa, they act as a catalyst each to other, and the efficiency of the coating increases dramatically. So certainly there is some interaction between nickel and rhenium in this solution, and it is of interest to understand this interaction. And here there are not very much, not many techniques that can actually do that, and we tried Raman for these purposes as well. First of all, as you can see here, Raman can be also used not for only for solid samples, but also for liquids and even for gases. And we tried many solutions, and finally we found out that there are actually 3 peaks on these characteristic shifts, which are responsible, they originate from different bonds of rhenium with oxygen atoms in the rhenium O4 ions. And for example, here on this graph, you can see that for different nickel concentrations, the ratio between 971 and 332 is different. And indeed, we were able to follow that for nickel, when nickel concentration is increased, the ratio between these 2 peaks is decreased. So it gives us an indication of which bond in rhenium O4 ion can be responsible for the interaction with nickel, and of course we are now going to proceed with this project and to investigate it in more detail. The next project I would like to present is with the research group of Dr. Shahar Richter, and they investigate adsorption of BSA, bovine serum albumin, doped with also other additions, other proteins, on gold-coated surfaces. So they take a chip of silicon, coat it with gold, then they anneal it in order to flatten it, and then they adsorb bovine serum albumin on this surface. Bovine serum albumin is a very famous protein. It is abundant in the blood, has also remarkable properties. Such self-assembled monolayers on gold, they possess interesting optical properties in the range from UV to near IR. So it is of importance to characterize such surfaces. Here you see the AFM image of the annealed gold. If you just coat the silicon wafer with gold, you will see something like that. It will be like granulated structure. So in order to make it flat, you need to anneal it. Here, as you can see, the annealing didn't work that much good, because you see some kind of walls and other residues on the sample. But still you can see that most of the sample is essentially flat. And after the absorption of BSA protein, you can see this picture, and first of all you see that the pictures differ significantly, which is a good indication. And also you can see that if you take, for example, line profile on the region where the annealed gold is quite flat, you see that the surface roughness here is less than on the BSA sample. And also on BSA sample we can see features which are quite small. For example, here we could see 30 nanometer. And if you take into account that the tip itself is about 15-20 nanometer, and the single molecule of BSA is about probably 10 nanometer inside, rough order of magnitude. So it could be that we see single molecules of BSA here, and of course, we need to continue with this research to make annealed gold sample better and further find the optimal parameters for the further characterization. The next project I would like to present is also connected to biological samples. This is in collaboration with research group with Dr. Natan Shaked. And they investigate human cells, which 10 minutes. Yeah? Okay, they investigate human cells which are normal and cancer HeLa cells, and it is quite hot direction now in applying AFM to such kind of research is to locally probe mechanical properties such as elasticity of these cells, and to find the differences, for example, from between normal and cancer cells, and by this to establish a new diagnostic tool for the early stages of cancer. So here you see the tip above the cell in just the optical image in the liquid, and we can take a force distance curve and take the calculate the elasticity using certain models in the software, and we used a different type of the probes for that. Initially, we just took cancer cells of different shapes and sizes, and saw that first of all we really measured the elasticity, the Young modulus, and also you can see here for the big cell it's about 1.4 kilopascal, and for other cells like this cell it's 2.2. Another cell gave 2.3, which are smaller cells. So it's not very statistical, of course. So we took more curves, but the main conclusion right now is that we do have an order of magnitude of the cell's elasticity, and the differently shaped and sized cells do have some deviation, some differences in elasticity, and the next step would be to compare it with the normal cells. Very similar research, actually also very interesting. Unfortunately, we obtained too much variable results, but still I would like to present it because it's a quite promising technique. And this is about biofouling. As you know, in the water pipelines, there are contaminations because of adhesion of microbes and bacteria to the pipelines. And the aim here is to investigate materials and probably treatments to these materials, to these surfaces, from which pipelines can be used. In the purpose to eliminate or significantly decrease this adhesion. For these purposes, we use functionalized AFM probe, where you can see 1 micron sized polystyrene bead functionalized with carboxyl groups, and this emulates the presence of microbacteria. And then we try different surfaces, different materials in air and in water, and see the adhesion of this probe to them by means of acquiring force distance curve. Here, for example, in air, you can see that for stainless steel, the force distance curve is very, very unusual. It indicates strong repulsion and even oscillation of tip, while for PVC, it is also unusual. Not like a regular AFM tip, regular sample. Here there is a strong adhesion. On water, both these surfaces exhibited adhesion. So this is quite remarkable. Unfortunately, as I mentioned, there was a very, very high variability in the further results. So I do not present them right now. I just want to mention that, for example, there are several reasons for such variability, such as surface roughness of stainless steel and that the bead was detached. I need to hurry up, so I will try to make it a little bit faster. The final project is a project with Professor Gil Rosenman. This is diphenylanin nanotubes, actually microtubes, called FF nanotubes, which possess very interesting properties, mechanical and thermal properties. For example, here you see that they are hollow, but then they close as a result of heating. They can be in linear or cyclic form, which is the conformation, the structure of a molecule, which can be distinguished by X-ray diffraction. And what was measured previously is that at temperature of about 150 Celsius, there is a transition from linear to cyclic nanotube, but it was not done at single tube level. Here we used Raman to acquire a spectrum from single tube, whereas we increased the temperature. This is the cyclic spectrum, linear spectrum. As you can see, during the heating up to 180 Celsius, different peaks, some of them grow, some of them decrease, some of them shift. Finally, the spectrum gets indeed closer to the cyclic, which is the further support of this finding at a single tube level. And I would like to thank to all these researchers that gave me opportunity to participate in their projects, and thank you very much for your attention.